Okay, today we're going to be talking about biotechnology. We're going to talk about DNA profiling, and we're going to be talking about genetically modified organisms. We're not going to be talking about cloning at this time. So what is biotechnology? Biotechnology, it can be defined as using technology for societal needs. As it says here, can be defined as the use of technology to control biological processes as a means of meeting societal needs. So what are some of the uses that we use biotechnology for? We use it for forensics, solving crimes. We use it for making genetically modified foods. We make it for transgenic biotechnology, which is also often used for pharmaceuticals and medical things. And we're not going to be talking about reproductive cloning today. Okay, so we're going to be talking about DNA profiling first. DNA profiling is very useful for forensics. You can prove guilt or innocence in crime scenes. You can establish paternity, so establishing family relationships, paternity and otherwise. Identification of human remains, so after such tragedies as 9/11 attacks on the World Trade Center, remains could be identified through DNA so that they could be returned to their families. Um, it was also useful during the times of in Argentina when they had a lot of people were disappeared, quote unquote, for reasons of their politics. Some of those children were adopted out to members of the military that were in control at the time. So the parents went to jail and were often killed. Their children were taken and adopted by the military, by different military families. And after that political upheaval was over, people wanted their grandparents wanted their grandchildren back. So they didn't really want their grandchildren to be raised by people who had killed their parents. So they were doing a lot of family relationship DNA processing in order to reunite those children with their true families. Also, species identification. So DNA has been used to track the sale of endangered species. You can find where an animal came from. For example, an elephant, you can find where that ivory, what band of elephants that came from by looking at different either DNA in the tusks, or also you can look at different mineral compositions to find out the food that they had gained and where that comes from. So DNA is very stable, and when it's saved in the right way, when it's stored properly, the stability of DNA allows crimes to be reinvestigated many years later. So this is just one instance of that. It took place in San Diego, and there was a case of an 8-year-old girl who was abducted from her home. She was sexually abused. The police accused her father, even though she claimed it was a stranger and not her father. The father was arrested when the child was convinced to change her story 2 years after the incident. But as the evidence was being gathered to prepare for the father's defense, they re-examined the girl's nightgown and found semen stains on it. And the semen stains they were able to get DNA from those stains, and they excluded the father and actually included a suspect who had been jailed on child molestation charges 6 weeks after the girl's assault, and so clearing the father. And this is an instance where DNA proves innocence, and it's often more important in proving innocence than in proving guilt, because it's easier to exclude somebody that was not there than to include somebody. You have to have a match for somebody to be a suspect. In other words, if you have DNA from the crime scene, but you do not have a suspect, then you can only wait until a suspect comes forward that you can match that DNA from the crime scene with. However, if you have a suspect and the DNA does not match, then it can exclude them and show that they were innocent, or at least not there at the scene. So it's very important. This is an example of a man whose innocence was discovered through the use of DNA. So he spent 17 years in prison. This man is Earl of Washington. He was accused of a murder he did not commit, spent 17 years in prison, but STR analysis, which is a DNA profile, we'll talk about how they're collected later, was used to overturn his conviction. And there's many of these innocent projects around the country now. And as of 1989, which was quite a while ago, 200 convictions were overturned. So there's been many more since then. It does take a very long time for the wheels of justice to turn in these cases. And many do not get the opportunity, but DNA is an excellent tool for using it to prove that people were not there and it couldn't have been them. This is another instance of DNA being able to be preserved under the right circumstances for a very long period of time. So this skeleton is 9,000 years old and it was found in Cheddar, England. So they call him the Cheddar Man. He's obviously not made of cheese, but he was found in Cheddar, England. So that's why he's called the Cheddar Man. And they got DNA from the bones or from the teeth, and this guy posing here with the skeleton is actually his ancestor. He's a school teacher, and they are separated by 300 generations, but they are related. You can kind of see it in the smile, I think. So how does DNA profiling work? The DNA analysis comes from non-coding regions of people's DNA. Most of our DNA is identical because we all make the same proteins. We have to make the same proteins to survive. But there are non-coding regions of the human genome, and these non-coding regions are highly variable. So these are the ones that are targeted for analysis. It usually involves some amplification, copying of the markers, and then the sizes of these amplified fragments are compared. Amplification is done by PCR. PCR, which we'll talk about in a minute, does the amplification. It takes a small sample and can make many copies of that to give you enough DNA to do an analysis. And then the sizes of the fragments are compared through electrophoresis, which we're going to talk about that process as well. Electrophoresis. This is probably, the writing on the slides is probably what makes these files too big. Okay. So what is done is, this is a gel. It's made of agar. And you get the DNA from the crime scene, and then you amplify it with PCR, making a little bit of DNA into a lot of DNA that you can test. You mix it with some restriction enzymes. These restriction enzymes will cut the DNA into fragments. These fragments will then travel down the gel. Because DNA is electronegative, it will travel towards the positive pole. And the larger fragments will not travel as fast as the smaller fragments. Just as we learned about in diffusion, smaller molecules diffuse faster than larger ones. So the larger ones will not travel through the gel as far, as quickly as the smaller ones will. And each one of these lines here on the gel will indicate fragments of the same size. So here we show the crime scene DNA being run, suspect 1 being run, and suspect 2 being run. And then if you compare the bands, all the bands have to match in order for there to be a match. So there is a match here, showing a complete match with suspect number 2 with the crime scene. So he had to be present at the crime scene. PCR stands for polymerase chain reaction, and it's a method of amplifying the specific segment of DNA. In other words, you take a little bit of DNA, you add some primers to it, and it will search for that little section of DNA that you want to be copied, and then it will start, the DNA will unwind and begin to copy itself. The sample is heated to separate the strands of DNA. We talked about how the base pairs in DNA are held together with hydrogen bonds. Those hydrogen bonds are a weak bond, and so they're easily broken with high heat. So you heat the DNA to help those strands break apart, and then you cool it with the primer, which binds to the target sequences, and then they're copied with a heat-stable DNA polymerase so that they'll be able to copy it even though it's in the higher heat. The advantages of PCR is that you can modify, you don't modify, you can amplify the DNA from a small sample. So you get a lot of DNA from a little tiny drop of blood or a drop of saliva, a drop of semen. You can get enough PCR, after PCR, you can get enough copies of it to do your DNA analysis. The concerns, of course, are that PCR will amplify any DNA sequence. So if there's contamination by even a small amount of DNA that contains those matching sites, so it has to contain the matching sites, but that's going to be a concern. So you have to be very strict in the way that you do the PCR. It has to be very, you have to guard against contamination and use very stringent conditions. Otherwise, you can get contamination. This is just a view here of how PCR works. You don't need to know the details about how it works, just the fact that it will take a single strand of DNA and make copies. So you can see here the first cycle yields 2 copies for each DNA. The second would yield 4. The third would yield 8. So each one copies over and over. And so in a short period of time, in about an hour, actually, you can get enough DNA to run a sample. So I did this over a summer school class once, and we PCR'd our own saliva, and they just ran it through a machine while we were at lunch. We came back and we had enough to do the sample. And then these DNA fragments are teased out. They're sorted by size by doing gel electrophoresis. The DNA sample is placed after it's PCR'd. You mix it with some restriction enzymes. The restriction enzymes are molecular scissors, and they will cut the DNA into fragments. Restriction enzymes. I'm just going to kind of write that in there. They equal the molecular scissors. Molecular scissors. So they cut out different fragments of the DNA. They actually look for repeating segments, and they cut at those repeating segments. And we're going to look at that in just a minute as how it does that. So then the DNA fragments are in different sizes, and then when you put them to the gel, the large fragments will not move as fast as the smaller fragments. So this teases out the different lengths of the fragments by diffusing them through the gel. And you can see which fragments are long and which are short. And each band that appears on that gel, like we looked at earlier, is going to be a collection of DNA molecules of the same length. So each one will be more than one DNA molecule, but they'll all be at that exact same length. So here's another view of it. You have the mixtures of the DNA here. You put in a power source. DNA is electronegative, so it runs towards the positive pole. As you put those samples in and you attach electrical current to it, they begin to flow from the negative to the positive end. And then you get to your completed gel, and you can see if you have a match. And here are these 3 fragments here, none of them match, because you have to be a complete match at all of the bands if you are doing a forensic test. If you're looking for paternity or something like that, of course, you're just going to match at some of the bands to show a familial relationship. But if you're looking for the exact same individual, they have to match at all of the bands. So STR analysis is the most commonly used type for DNA profiling. STR stands for short tandem repeats. And these are genetic markers that are used. Short tandem repeats are repeated sequences in people's genomes. The number of repeating units can differ in individuals. There can be up to 80 different variations within a human population of one particular site. So the standard now is, I think it's gone up from 13. I think it's more like 16 or 17 different sites now. And you have to be a match at all these sites in order to be a perfect match. So what's an STR? Here's an example. Chromosome 7 is one of the 13 standard loci used. Its sequence is GATA, so it can be repeated from 5 to 16 times. So GATA, 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 GATA. I might have that in my genome like 12 times. You might have it in yours 5 times. So this is going to result in longer fragments. If it's only repeated 5 times, it's going to be a much shorter fragment than if it's repeated 12 times. There's 78 possible different genomes in this one loci. So if you look at 13 or 16 different STR sites, you're going to see that pretty quickly you can narrow it down to one particular human. So how reliable is DNA profiling? With only 13 STR sites, the probability of finding the same DNA profile in 2 unrelated individuals is less than 1 in 10 billion. So there's over 7 billion people on the planet, so it's pretty easy to see that you're going to match it down to one individual, especially one individual that could possibly have been at the crime scene at that time. So it's not foolproof, of course. The science behind it is irrefutable. However, you could have insufficient data, you could have contamination, human error, you could have flawed evidence, you could have planted evidence. So it's not perfect, but if you do have good DNA, and like I said, the science behind it is irrefutable. So if nothing went wrong with the collecting of it, the storage of it, or the processing of it, then your results are very reliable. It's, of course, not as quick and easy as they show on TV. It usually takes many days or weeks for it to be completed. So you can't do it in a commercial break. So this is another example of it here. You see this, here's one site, it's AGAT. So each one of these yellow boxes here is AGAT, 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 you get the idea. So the enzymes, restriction enzymes will look for this sequence and cut it at these areas after the AGAT stops. It will cut it. And this is called a short tandem repeat. Here's site number 2. The short tandem repeat is GATA. So that's repeated many times here, not quite as many times. So it matches at site 1, but it's different at site 2 because this one down here is much longer. So if you run these in a gel, this is what you're going to get. Site 1 matches and site 2 does not. So you already have a difference. With just 2 sites between these 2 individuals, you can tell that there's a difference between the 2 individuals. So these a single nucleotide polymorphism or an SMP can change the length of these short tandem repeats. The short tandem repeats, if you just change one base pair, which is a single nucleotide polymorphism, because this is a nucleotide here, and that's just a single one. Changing a G to a T, for example, will cause the restriction enzymes to cut at a different place. They'll just simply skip over it and cut at a different place, making for a different length fragment. So an RFLP is a restriction fragment length polymorphism. So what does that mean? Restriction fragment, when the restriction enzymes cut it, it becomes a restriction fragment. And then the lengths are polymorphic. So the lengths are polymorphic. Polymorphism means different morphs or different sizes, different shapes, whatever. So they look different, and then the lengths are different. So length polymorphism. So restriction fragment from being cut with restriction enzymes, leave fragments of DNA, and then their lengths are polymorphic. The lengths are different. So RFLPs are the result can be the result of the single nucleotide polymorphism. So you change one nucleotide, and when the restriction enzymes go to cut the DNA, it cuts at a different place and thus makes it a different length. So we can look at that here. So the restriction enzymes added, and they're looking for these little, they do a little dogleg cut because DNA runs in that anti-parallel fashion. So you got CCGG here and CCGG going in this direction and in this direction. So what it's looking for, it's cutting between the C's. When it sees a CCGG, it cuts between the C's. So you see there's a cut here and a cut here. This one, because of that single nucleotide polymorphism, that's an N, right here. It didn't see the CC there, so it didn't make a cut. It makes the cut down here. And then when you put these two fragments into a gel, you're going to get very different results here. Sample number 1 is going to give you X, W, and Y, whereas sample number 2 is only going to use Z and Y because it didn't have that cut up here due to that SNP, that was that single nucleotide polymorphism that was different in this individual's DNA. So since DNA is so accurate, how do you think it compares with eyewitness identification? So this is something that we'll talk about when we Zoom. And in practice, DNA profiling in forensics can be said to be more effective in proving innocence than in proving guilt. So this is another topic that we can discuss when we Zoom. The reason for that is because it's easier to exclude than to include. And eyewitness, we'll talk about the problems with eyewitness identification. Okay, genetically modified organisms. Genetically modified organisms are organisms that have had genes from other organisms inserted into their genomes by artificial means. This is a lot different from selective breeding. Obviously, we've been doing selective breeding for centuries in order to get organisms, animals, plants to produce large fruits or to produce more milk or to produce leaner meat, whatever we're looking for. Simply through breeding them, selective breeding, but this takes it to a whole new level where we actually take a gene, and often the gene is from an unrelated organism, and implant it directly into the genome rather than breeding an animal to get a new set of genes. So genetically modified organisms are becoming a lot more common, especially here in the United States. We have a lot of agricultural products that are genetically modified. Some countries kind of ban them outright, like France. They do have benefits. They do have some potential problems as well. So it's a matter of weighing what the benefits are against the potential problems, and going forward with this technology as it becomes more and more common, we need to think about some of the potential problems as well as just the benefits. There are both. So one of the potential problems is that the genes in a closely related organism can spread. So for example, if I was to take radishes and put in a gene that keeps them from being attacked by a certain pest, So they are now resistant to that pest. If I then have wild radishes that grow within the vicinity of my cultivated radishes, because plants transfer pollen easily from one plant to another because they use vectors such as bees and other insects. So the pollen from my genetically modified radishes might get into the population of non-genetically modified radishes, and those would be into the, I should say, into the population of wild radishes. Wild radishes happen to be a very invasive plant, so this makes an invasive plant even harder to kill. So for instance, if I put in genes that would make it resistant to a pesticide, not a pesticide, a herbicide, then that wild radish, which is an invasive species, would then be resistant to that herbicide as well. So it might be harder to eradicate. Another problem is allergens. It may insert allergens into a food supply, these genetically engineered genes, and that could be problematic, and it's not always easy to know that before something hits the market. So the FDA requires evidence of safety before approval, but a lot of these things aren't tested for a very long period of time, so we're not entirely sure of long-term effects. One of the issues that has been a problem in the past is with BT corn. BT corn is a genetically modified corn that has a pesticide in it that kills the BT corn borer. So this is a moth, and it lays its eggs inside the corn. They develop into larvae and they eat the corn from the inside out. It causes a lot of damage to corn. So they came up with a genetically modified corn where it has this toxin in it that the corn actually produces itself. Rather than spraying it on the corn to kill the corn borers, the plant itself produces it. And this has led to some problems, though, because one of the things, I'll get to this in just a second, one of the things that it does is it also kills other lepidopterans. So moths are lepidopterans. If it targets a particular moth, it may also kill monarch butterflies, which are also lepidopterans. So getting back to this part here, the idea behind this, I'm actually going to insert another slide so I can show you what I'm talking about. Okay, so the idea here behind the refuge plot, if I was to grow BT corn in this plot here, and then I have a refuge plot of non-BT corn, so the idea is that farmers have to plant refuge fields. So this would be the refuge field. The idea is that the corn borers coming in contact with the BT corn in this field here would, over time, they will begin to develop a resistance to the BT corn, just as they would develop a resistance to being sprayed with a pesticide. So this genetically modified process has not eliminated the fact that the organisms that are targeted will develop a refuge. So it will develop resistance. So the idea is to have a refuge field for them so that these guys over here, so these corn borers, don't become immune to it. They don't build up resistant. They don't build up a resistance. So they don't become resistant. And then because they don't become resistant, they breed back with the ones that are resistant, lowering their resistance. Is there anything wrong with this idea? Pause for emphasis. Yes. hopefully you see the problem. Um, Genes will make a two-way street. So you can't say that this doesn't happen, that actually confer the resistance on the non-resistant corn borers rather than the non-resistant corn borers giving their non-resistance to the resistant corn borers. You get what I'm saying? In other words, the genetics is going to flow both ways. So this is not a viable solution. This is just silly to think that you could create a one-way path of genes, of gene flow in a population. Okay, and as I said earlier, the other problem with this is that anything that targets one species of Lepidopteran may target another species of Lepidopteran. So they did a laboratory study. These are monarch butterflies, and they're protected by law. A laboratory study showed 50% mortality for larvae of the monarch butterfly feeding on leaves of milkweed plants dusted with the Bt corn pollen. So, of course, the pollen blows in the wind and will land on other plants. And then they did a field study planting with plants at various distances from the Bt cornfield, and they showed a 19% mortality for the monarch larvae feeding on plants closest to the field. So the take-home story of this is, of course, that it just as spraying pesticides on plants killed non-target species, so does genetically engineering plants. It also kills non-target species. So we haven't eliminated that problem with the genetically modified organisms. This is a lab in Paris to keep us all safe from genetically modified organisms. As I said, they kind of ban things in France. They don't trust it in the agricultural products for humans. Benefits. This is supposed to be a slide here showing genetically modified organisms. I guess I should switch the color here. I can't see the purple on there. Genetically modified and non-genetically modified. I think this is corn in a grass field, but at any rate, it still illustrates the fact that, of course, you're going to get increased yield. You can get increased nutritional value. This can be very important. Increased hardiness. This can be very important. Increased yield. Of course, that leads to more money. One of the big problems is a lot of these genetically modified organisms have been done by a company called Monsanto, and they're genetically modifying plants to be resistant to Roundup so that you can dump lots of Roundup onto your agricultural fields and it won't kill the plants that you want, the plants that are your crops, your money crops, but it will kill weeds. This is not really a good thing to do because Roundup is toxic for the environment. So increasing yield, increasing nutrition value, increasing hardiness, these are all very worthwhile causes, and sometimes they are more important than the possible downsides of making a genetically modified organism. Increased nutritional value, this is done with golden rice. See, yes, that's the next slide I put here. Golden rice was one of the first genetically modified organisms, and what it is, it's a transgenic variety. Transgenic means genes from a different organism. So they took some daffodil genes and put them in with rice. So they contain beta carotene now, which our body uses to manufacture vitamin A. And vitamin A deficiency, it causes blindness in developing children. And so this is a real problem in a lot of developing countries. So this genetically modified rice could cure that problem. However, it was not seen as a good idea by a lot of the countries that we tried to market it in, and I think it's starting to be effective now. They actually have a second variety out which has much more beta carotene in it, so it's giving out more vitamin A, and I think they're going to start growing it in some Asian countries. So this is a good use of genetically modified organism. Before we go on to transgenic organisms, I just wanted to talk real briefly about the only genetically modified organism to date that is an animal, and it's a fish. And it doesn't look like this because this is my little hand-grown fish here, but it's salmon. So a genetically modified salmon after a year would look like that. A wild salmon after a year would be significantly smaller. So the problem with genetically modified salmon is if they get out into the population and they grow really, really rapidly. This is the GMO. This is an exaggeration, of course. This is the wild guy. The problem that they foresaw was that as the GMO gets out into the wild, which it's not supposed to because there's only supposed to be farmed in pens, but if they got out into the wild, then they might outcompete the wild salmon. However, when they put them both in streams to test this, the wild salmon outdid the GMO salmon. I guess they're just tougher and know how to live in that type of an environment. So we'll have to wait and see if this is a bad idea or not. But they have a genetically modified salmon, and they've been modified with genes to make them grow very rapidly. Okay, let's move on to transgenic organisms. Transgenic means that it contains a gene from another species, at least one gene from another species. So transgenic organisms, they often use a vector. E. coli is one of the vectors that are often used to transplant a gene into another organism. So the gene is put into the E. coli genome and then can be transferred into another organism. Or it can just be transferred into E. coli and grown in E. coli. So human growth hormone, for example, is produced within a bacterium, and that's been made transgenic by importing a human gene. So you could put a human gene into E. coli, grow lots and lots of E. coli in a vat, filter it out. This is my filtering process, and you end up with your hormone. It's much cheaper. You can make much larger quantities of it, and you can really control the purity of your hormone, of your product with this. So it's a really good way to make pharmaceuticals. This is human insulin that's also being produced in bacteria. So these recombinant organisms are used. So if you can do it in a prokaryote, often E. coli is used. If you have to use a eukaryote, then Saccharomyces cerevisiae brewer's yeast is often used. So you can put the gene into the bacteria and grow the bacteria in big tanks, and then you collect it, you purify it, and you get your product. Or if you can't use a prokaryote, then you can use a eukaryote, like Saccharomyces cerevisiae. That's a yeast, and so if the cell won't grow, if the product won't grow in a bacterial cell, a prokaryote, sometimes you have to use a eukaryote to grow it. And so the prokaryote has a big advantage because of its rapid reproduction, and it's inexpensive. A eukaryotic host can be used. Again, yeast is the fastest and easiest because of rapid reproduction. They reproduce by budding. They don't have sexual reproduction, so they're very quick, just like E. coli, which reproduce by, I forgot the term. Ugh, we just learned it too. Just duplicating their genome and then elongating the cell and breaking it in half. Some products can't be made unless you put them into an actual mammalian cell culture, or you can also use farm animals. So farm animals will secrete the gene in their milk, which can then be filtered, and the protein can be taken out, whatever protein that is. So these are some farm animals. These are some sheep, and they carry a gene for a human blood protein that's being tested for use in cystic fibrosis. I'm sorry, for use in treating cystic fibrosis. The protein is harvested from the sheep's milk. Other examples include goats that produce tissue plasminogen activator, TPA, for heart attack victims, sheep that produce alpha-1 antitrypsin for treating emphysema, pigs that produce hemoglobin as a blood substitute as well. So there's a lot of these transgenic farm animals that are producing human proteins that can be, again, it's a lot cheaper, faster, and you can really control the consistency of the product. This is just a chart showing you all the different types of products that we now get transgenically, and this shows what they're made in, and this shows what they can be used for. So this is just reiterating the same thing. You can get insulin, you can get human growth hormone to treat dwarfism, insulin treats diabetes, of course. You can also use these transgenic pharmaceuticals to diagnose and treat diseases, testing for inherited diseases, and detecting infectious agents such as HIV. We might even come up with something for COVID-19, who knows. What I wanted to tell you from this slide is what a vaccine does. A vaccine works by stimulating an immune response by injection. Again, hopefully we'll be getting a vaccine in a year or two that will fight COVID-19. But what is usually used is usually a protein from the surface of the infectious agent or harmless version of the infectious agent, and then it's injected into the bloodstream. The body will recognize those proteins, and that's when it comes in contact with the actual disease. It will already have that that army that it can call up, that can combat the disease when it faces the real thing, because it will recognize it. So that's the idea behind a vaccine, to stimulate an immune response. And we'll talk a little bit more about that when we do the Zoom on it. We can talk about more examples. So the overall advantages of recombinant products are you can get a human protein, you can get good purity, good quantity, and less expensive as well, of course. Less expensive, because anytime you have something in larger quantities that can be produced quickly, it's going to be less expensive than the old ways of doing it. So genetically modified and transgenic organisms, these are reproduced by artificial means. They are by taking a gene and inserting it into the genome of the host. You can, for genetically modified plants, you can make them resistant to herbicides, resistant to pests, or increase their nutritional profile or their hardiness. For genetically modified animals, you can improve the quality of the product, make more of it, the quantities, and it's really good for the production of therapeutics, pharmaceuticals. The way it's often done is with inserting it into a plasmid. So you can insert the gene into a plasmid. Plasmids are these extrachromal amounts of, here's the chromosome of the bacteria. We can call this one E. coli. This is a plasmid. This is easily transferred to another bacteria. We'll talk more about how that's done. So let's say this one there's a little gene on here that we want to transfer, and that gets transferred either into the plasmid of the other, sorry, that was supposed to be red, the plasmid of the other bacteria, and then every time that bacteria reproduces, it's going to be reproducing that same section of gene, or it could be transferred into the main chromosome area. And again, either way, it's every time this particular bacteria replicates, it's going to replicate that particular gene that we transfer. And we probably won't be doing that, so we are done for today.